Okay, perfect. So I'll try to be fast. Thank you for the opportunity. I tried to use it more to present a kind of doubt problem that I had specifically when applying POMO models, more than any actually biological insight from this system. So hopefully to find someone who to discuss with this along the workshop. So anyway, the project is on phylogenomic analysis of cone shells from, in this case, from Cabo Verde, West African islands, around here. And needless to say, there's a lot of people involved, from the fieldwork experts to us doing more genomic analysis. So, so this group is highly diverse with a huge distribution, depending on the classification, which they vary a lot, but this can be considered a single genus. And of course, things vary a lot in shapes and color patterns, and then they tend to be classified based on that. So, of course, since along the last decades and with molecular data, species diversity, real or not, increased a lot. Both for the full genus as both for the small geographic radiation, let's say, in this case, the Cabo Verde one. And with most of the data being mtDNA, like single genes or some genes or mitogenomes, and most of the species delimitations being based on some kind of divergence threshold method, like ABGD or BBB, or even only on a kind of percentage of mtDNA divergence. So what we wanted to do was to give a more genomic perspective to this thing. So very shortly, we from raw sequencing of 2 exemplars from the from each of the Cape Verde variation, we used anchored hybrid enrichment protocol and ended up isolating roughly 1,700 loci, anonymous loci, and doing a target capture experiments with loci of different sizes from almost 2000 BP to 500, and with these being the distributions of variable and parsimony formative sites. So jumping quickly, I jump over these. This is just to show you a bit of this particularly small group shape, color variation thing. Jumping a bit over this, this could be the kind of current sampling, and what we asked to the experts that actually know how to recognize these things or that classify these things was, sample us the things that you recognize, that you could recognize, is that you think that can be different things, that you would call a different morphotype, or that you think they can have different names. And they ended up with so around 16, let's call, populations, okay? And taxonomy meanwhile changed, and now like 5 species within 3 mitochondrial clades here represented by colors are defined. Yeah, so very quickly, when we look at mtDNA trees, we see 3 clades, as I said, and the current species classification is based on mtDNA. So you see these 1, 2, 3, 4, 5 things. When you look at concatenated nuclear 3, you see some evidence of some integration and some discordances, especially the main order of divergence of the main clades is completely different. So here the C goes out first, and here the A goes out first. So we explored species tree inference methods. Well, to define the species or populations, or whatever you want to call it, we used the SNPs from this data, the BLLX SNPs in this case, and the structure approach. So basically, we inferred these clusters, and we did a nested analysis at every step, removing individuals with sign of integration and dividing as far as we could. So we ended up with kind of 16 population units that we defined as our tips for species tree analysis. So then is the, the funny thing that I would hope to discuss with someone along this day. So if you look at the astral tree, it gives you this topology, so this ABC, with very well supported, only branches with local PPs. Oops, sorry. Only branches with local PPs above 1 are noted, all the others are fully supported, despite the size of lineage sorting or maybe other factors. But almost every loci subsampling things, and we tried many, gives us this result very well supported. While I tried RevPoMo also, because supposedly it performs well. Well, I guess it performs well on shallow things like this. So these mtDNA things are tops 2%, 2 point something, 3% divergence. So it's very shallow phylogenies. And the thing is that with RevPoMo, result is completely different with in this case with the C. So in this case, the A pops out first. In this case, the C pops out first. There's other things, but these are kind of the main. And they seem very well supported also. And I tried several virtual population sizes. And well, this was one done with all the sites, which I think is the way to perform it. And yeah, anyway, this is just kind of intriguing, and that was actually what I was hoping. We are exploring why this can be, and the influence of possible influence of gene flow and deep coalescence in the whole system. But I was hoping to find someone or to learn something more about this to have a better idea. Thank you.